Now in this video I will explain on the next group of polysaccharides, okay, that is heteropolysaccharides. Now heteropolysaccharides, they are also referred to as the heteroglycans. They have two or more different monosaccharides, okay, linked together to form a polysaccharide, and usually these repeating units of disaccharides, although in some cases the polymer it has two alternating disaccharides. So these are the repeating units of disaccharides, okay. Now some important heteropolysaccharides, they are the Glycose Aminoglycans, Pectin, and Agar Agar. So we are going to discuss about these three. Now coming to the Glycose Aminoglycans, the polysaccharides that are composed not only of a mixture of simple sugars but also of derivatives of sugars such as amino sugars and also uronic sugars. Those type of sugars, they are referred to as glycose aminoglycans or also mucopolysaccharides, okay, they are also known as mucopolysaccharides, and they are gelatinous substances of high molecular weight, up to about 5 into 10 to the power 6. So they have a very high molecular weight, and most of these, they act as structural support material for the connective tissue or mucous substances of the body, and they serve both as a lubricant and a cementing substance. Now some of the mucopolysaccharides are also found to be present in combination with proteins, thus forming mucoproteins or also known as proteoglycans. The proteoglycans, okay, where these polysaccharides are found in combination with proteins, are known as proteoglycans, and the proteoglycans, they usually consist of 95% of carbohydrate and 5% of proteins, okay. So some of the important glycose aminoglycans are given here, okay. Chondroitin sulfate, Keratin Sulfate, Hyaluronic Acid, Heparin, and Dermatitis Sulfate. Now chondroitin sulfate, if you see here in this figure, it is actually a repeating unit of disaccharide of beta-D-glucuronic acid and the N-acetyl galactosamine 6 sulfate, okay. The anomeric carbon of the glucuronic acid is linked to carbon number 3. Here you can see this is the carbon number 3 of the N-acetyl galactosamine 6 sulfate. Now the repeating disaccharides, they are linked by the beta-1,4 linkage, and this chondroitin sulfate is found in cartilage. So the next important glycose aminoglycan is Keratin sulfate, which has a repeating unit of disaccharide beta-D-galactose N-acetyl galactosamine 6 sulfate, okay. And these units, they are linked by N-acetyl galactosamine 6 sulfate 1,3-beta-D-galactose. So this keratin sulfate, it keeps the cornea transparent, okay. The function of this saccharide is keeping the cornea transparent. Now another important polysaccharide which fall under this category is hyaluronic acid, and this one is just like the chondroitin sulfate. It differs from it in not having a sulfate in galactose. Here you can see in this N-acetyl galactosamine, the sulfate is absent. Thus in beta glucuronic acid, N-acetyl galactose, it is referred to as the beta D-glucuronic acid 1,3 N-acetyl galactosamine. Okay, so this, because the link here, the glycosidic bond is between the carbon number 1 and carbon number 3. So the difference is just in this case where in chondroitin sulfate we have a sulfate group in carbon, in this N-acetyl galactosamine, whereas here it is absent. Now this particular polysaccharide hyaluronic acid, it serves as a lubricant as well as a shock absorbent in the joints. Okay, so coming to the next important heteroglycans or heteropolysaccharide is heparin. Okay, this heteropolysaccharide, it has the repeating unit of the disaccharide L-iduronic acid 2-sulfate and the N-sulfoglucosamine 6-sulfate, and these two, they are linked together by the alpha-1,4 linkages. The L-iduronic acid monomer here, as you can see, it has the sulfate group in carbon number 2. Alright, it has the sulfate group which replaced the OH group at carbon number 2, whereas the glucosamine, it has the acetyl group replaced by a sulfate. You can see here where the acetyl group it has replaced by the sulfate group. Now the another sulfate, it replaced the OH group also in carbon number 6. Here we usually have the CH2OH group, but it has replaced the OH, the hydroxyl group has already been replaced by the sulfate group. So this is the structure of heparin. Now coming to another important heteroglycan is dermatin sulfate. Now dermatin sulfate, it resembles closely to chondroitin sulfate. As you can see here in this figure, these two, they are similar, and was therefore this dermatin sulfate, it was earlier described as the chondroitin sulfate B because of its similarity with the chondroitin sulfate. However, if you see here, it differs from the chondroitin sulfate in having the L-iduronic acid in place of the D-glucuronic acid as the first unit of the disaccharide. So the second unit here is N-acetyl galactosamine, which is sulfated at carbon number 4. You can see here, this is the carbon number 4. The N-acetyl galactosamine is sulfated at carbon number 4, where the OH group is replaced by the sulfate group. And these two units, they are linked together by the beta-1,3 linkages. You can see here the glycosidic bond is formed between the carbon number 1 of one residue and the carbon number 3 of another residue. And the repeating units, they are joined by beta-1,4 linkages. Just remember this, that the repeating units of these disaccharides, they are linked by beta-1,4 linkages, whereas these two monomers, they are linked by beta-1,3 glycosidic linkage. The here heparin molecule initially, it is synthesized in a non-sulfated form, whereas later it is deacetylated and the acetyl group is then replaced by a sulfate. So here in heparin molecule, the sulfonation, it may not be complete, and some of the residues present in heparin, they may remain unsulfated, and in most molecules, therefore the degree of sulfation, it may vary, and this variation, it may involve both the position, that is the N sulfates and the O sulfates. So all those, they are glycose aminoglycans, they fall under glycose aminoglycans. Now coming to the another important heteropolysaccharides, that is pectin and agar agar. This is the structure of pectin. Pectin is actually the important cementing material in plant tissues, and it is also the main constituent of the middle lamella. This polysaccharide is this pectin is a polysaccharide of the alpha D galacturonic acid joined by alpha 1,4 linkages. Okay, it is joined by alpha 1,4 linkages, and it is not included in homoglycans because some of its glucuronic acids, they are methylated esters, as you can see here. Okay, some of the galacturonic acid units, they are methylated esters, and the degree of esterification, it increases the gelling property of pectin. So pectin therefore is regarded as an important constituent of jellies and jams. Now coming to another important heteropolysaccharide, that is agar agar. This polysaccharide, heteropolysaccharide, is extracted from seaweeds, the members of rhodophyceae like Gelidium, then Gracilaria and Campyliflora. And as you can see here, that the repeating disaccharide unit agar agar is known as agarobiose. Okay, it is known as agarobiose. So the agarobiose here, it is composed of beta D galactose and 3,6-anhydro beta L galactose. These two monosaccharides here, they are linked by a glycosidic bond between the carbon atom number 1 of the beta galactose and also the atom carbon number 4 of 3,6-anhydro beta L galactose. And these repeating units, they are joined with another repeating units through beta 1,3 linkages. Okay, so this is the structure of agar agar. Now coming to another very important heteropolysaccharide, that is glucomannance. Now this glucomannan is a fibrous polysaccharide with which is a strong absorbent, and about 1 gram of this particular polysaccharide, it can absorb up to 200 ml of water, and hence this polysaccharide is used in making disposable diapers and sanitary napkins. This polysaccharide, this sugar, it is made up of glucose. Okay, it is made up of glucose and mannose units. Okay, it is made up of these two monosaccharides, which may be in the ratio of 5:8. The ratio of glucose is to mannose is 5:8, and these units, they are usually joined by the beta 1,4 glycosidic linkage. And the main chain, it may also have side chains. As you can see here, this is the main chain which is joined by the beta 1,4 linkages, and it also has a side chain. and these side chains of around 11 to 16 monosaccharides, they usually occur after every 50 to 60 units. Okay, these branches or these side chains, they occur after every 50 to 60 units, and these branches, they may be attached to the main chain by beta 1,3 linkages. As you can see here, that the anomeric carbon of this residue, okay, the carbon number 1, it is linked with the carbon number 3 of this main chain, the carbon number 3. So the glycosidic bond which is formed here is referred to as the beta 1,3 linkages, and about 5 to 10% of the glucose units, they may have acetyl group attached to carbon number 6. You can see here in the second glucose residue here, this is a glucose residue. The second residue here, it is the acetyl group is attached to carbon number 6. And also the distribution of glucose and mannose units does not follow any definite pattern, and they may be present in blocks of glucose or mannose, or even alternate in some region. However, the common pattern is usually glucose, glucose, mannose, mannose, glucose. Okay, so this is the common pattern in, or we may have all the repeating units. Okay, we may have all 5 mannose residue followed by the glucose, glucose and mannose. So this is the usually the common pattern. But the main chain, it may also have the acetate groups in carbon number 6 after every 9 to 19 residues. Okay, as you can see here in residue number 2, in glucose residue, the acetyl group is present at carbon number 6. So this may repeat after every 9 to 19 residues. And the hydrolysis of this acetate forms intermolecular hydrogen bonds. Okay, it forms intermolecular hydrogen bonds which causes gelling. So these are all the important heteroglycans or heteropolysaccharides. Now I just want to explain a little bit on the another types of saccharides, that is conjugated, another type of glycans, that is conjugated glycans. These conjugated glycans, uh, they are those molecules which are found in association with other molecules other than carbohydrates. Okay, so there are a large number of carbohydrates, the large heteroglycans or oligosaccharides, which are found in association with molecules other than carbohydrates, and these molecules can be either lipids or proteins, and these conjugated forms, they are known as conjugated glycans or uh, pro and they are of 3 types. Okay, that is a proteoglycan, glycoproteins or glycolipids. The proteoglycans, these compounds, they are the complex molecules which are made up of proteins covalently linked in glycose aminoglycans. Okay, the proteoglycans, they are the complex molecules made up of proteins linked covalently in glycose aminoglycans, and about 95% of the complex is carbohydrate. The majority of the glycose aminoglycans is in the body they are linked to the core protein forming the proteoglycans, or also known as the mucopolysaccharides. Okay, they are also known as mucopolysaccharides. Now these glycose aminoglycans, they are attached at right angle to the plane of the core protein. So one of the example of proteoglycans is Agrican. Now this Agrican, it is one of the most important proteoglycans which are present in the extracellular matrix, and each Agrican molecule, it contains many chondroitin sulfates and keratin sulfates. So the bacterial cell wall also, it has a chain of repeating disaccharide units made up of N-acetyl glucosamine and N-acetyl muramic acid, which are joined by beta-1 linkages. These also, they are example of proteoglycans. And the next category, the next group of conjugated glycans are glycoproteins. These glycoproteins, they are small oligosaccharides, okay, which function as labels or signals in carrying the large protein or large lipid molecules to their destination, thus making a glycoprotein when used to mark a protein, and glycolipids when used to mark a lipid. Okay, so these glycoproteins, they are ubiquitous in nature and they are present in the cells. Either they are bound to the membrane or they are present in the extracellular matrix. And the important function of the carbohydrate component of the glycoprotein is to provide specificity to the protein molecule. And so in this figure you can see the N-acetyl glycosamine. Through this linking polysaccharide, you can see the binding of the N-acetyl glycosamine to the core protein. So this is one of the example of a proteoglycan, and this is another example of the peptidoglycan of the cell wall of bacteria. As you can see here in this figure, that the bacterial cell wall, they has a chain of the repeating disaccharide units. Okay, repeating disaccharides units here made up of the N-acetyl glucosamine and N-acetyl muramic acid, which are joined by the beta-1 linkages. And these chains, they run parallel in the bacterial wall, and they are linked by a peptide chain as well. Okay, they are linked by a peptide chain, as you can see here. And each N-acetyl muramic acid residue, it has a tetrapeptide. The L-alanine, these four peptides here you can see, okay, these four peptides, the L-alanine, D-glycine, L-lysine and D-alanine, link to the carboxylic group of the lactic acid. And here you can see link to the carboxylic group of lactic acid at carbon number 3 here. And the third amino acid residue, the L-lysine, in this case this third amino acid residue of this tetrapeptide is then again linked to the fourth amino acid residue, D-alanine, of the adjacent acetyl muramic acid in the other chain, okay, in the other chain by a pentaglycine, here L-glycine chain. Thus the pentaglycine crosslinks, they bind to the heteropolymer chain to provide more strength. So this is the structure of the peptidoglycan of the cell wall of bacteria. So the example of the glycoproteins, some of the examples are the immunoglobulins. Okay, and also the major histocompatibility complexes such as the egg white, the blood plasma, and a number of hormones. These are all several hormones, immunoglobulins, blood plasma, egg white, all these they are example of glycoproteins. Now we come to the next group of conjugated glycans, that is the glycolipids. Now the carbohydrate moieties may also join with the lipid molecule, and several of them they are the important constituent of the cell membrane. These the simplest glycolipid known is cerebroside. Okay, it is a cerebroside which contains a single sugar residue, that is either a glucose or a galactose, and it is derived from sphingosine where the amino group is linked to a fatty acid by an amide bond. Okay, these another examples they are the gangliosides. Now gangliosides they are more complex glycolipids compared to the cerebrosides because they contain a branch chain of about 7 sugar residues. And another example is Globocyte. Globocyte is also a glycolipid with 2 or more sugar, usually D-glucose, then we have D-galactose, and also N-acetyl D-galactosamine. And these glycolipids, they also function as determinant of the blood group. So this is the structure of cerebroside. Okay, this is the structure of cerebroside. As you can see here, the in cerebroside, the in this glycolipid, that is cerebroside, this is derived from a sphingosine where you can see here the amide group. Okay, the amino group is linked to a fatty acid. You can see this one here. This one is linked to the fatty acid by an amide bond. And the terminal hydroxyl group is linked to either a glucose or a galactose. In this case, you can see it is a galactose molecule by a glycosidic bond. And another example is of a lipopolysaccharide, that is a glycolipid in the outer membrane of the gram-negative bacteria E. coli or Salmonella typhimurium. They has a lipopolysaccharide where six fatty acids, they are joined to two. You can see here they are joined to two glucosamine residues, okay, which are themselves joined by this bond here, which is the alpha 1,6 linkage. So these two glucosamine residue, they are joined by alpha 1,6 linkage, and there are 6 fatty acids here, these 6 number of fatty acids which are joined to these two. And the carbon number 4 here of the first residue, all right, this carbon number 4 of the first residue and carbon number 1 of the second residue, they are phosphorylated. And the carbon number 6 of the this carbon number 4 and this carbon number 1 of the first and the second residue respectively, as you can see, they are phosphorylated. Okay, and the carbon number 6 of the first residue here, it is also linked to a large polysaccharide, and this polysaccharide chain involves about 50 to 60 residues, okay, representing 8 different monosaccharides.